So let's talk about gases. Again, going back, oops, we're talking about the things that influence microbes. So, oxygen actually is very impactful for microbes and their growth. They kind of fall into three categories when it comes to oxygen: those that use oxygen, which means they can detoxify it; those that neither can use oxygen nor detoxify it, okay? And those that do not use oxygen but can detoxify it. So I've got, I need it, I need to be able to use it. I don't need it, but it's okay because it's not going to hurt me. And I don't need it and it can kill me, okay? So the problem: oxygen can be transformed into one of several toxic byproducts. Singlet oxygen, superoxide ions, hydrogen peroxide, hydroxyl radicals. Microbes that can grow in the presence of oxygen actually have to detoxify all of these products using special enzymes. Superoxide dismutase is going to be an enzyme that basically converts the superoxide ion into hydrogen peroxide. Now, do you drink hydrogen peroxide? No. Why? Because it's poisonous. So they've got catalase. We have catalase, so a little bit of hydrogen peroxide isn't going to hurt us, but a lot would actually poison us. So catalase is an enzyme that basically takes hydrogen peroxide, H2O2, and breaks it up into water and oxygen. Okay? So those that can do any detoxifying have these guys. Okay? So, aerobe. Aerobe, that word, okay? Always think oxygen, okay? So, an aerobe uses oxygen in its metabolism. It possesses the enzymes needed to basically process the toxic oxygen variations. An obligate aerobe has to have oxygen to live. We are obligate aerobes. We have to have oxygen to live. Okay? It's not like I can put you in a vacuum and you'll do great. It's not going to work. You have to have oxygen. So, examples of aerobes include most of the fungi. Notice I didn't say all. Most protozoans, many of the bacteria, including Bacillus species and Mycobacterium tuberculosis. Where does tuberculosis affect you? Your lungs. What's in your lungs? Oxygen. So they like your lungs because the oxygen that they need, that they have to survive on, is there. All right, microaerophile. Okay, phile means loving, micro, small, aero, oxygen. It does require a small amount of oxygen for metabolism, but normally it doesn't grow in our atmosphere's concentration of oxygen. So I said oxygen is about 20% of our atmosphere. Yeah, that's going to be too high for these guys. So, two examples of microaerophiles are Helicobacter pylori, that's the one that causes ulcers in your stomach. You don't have a ton of oxygen in your stomach. And Borrelia burgdorferi. There we go. Facultative anaerobe. Okay, facultative. To facilitate means to kind of help along. They don't require oxygen for metabolism, but they can use it when it's present. If it's there, I'll use it, basically. They grow better in the presence of oxygen, but they can live with it or live without it. Okay? Minigram negative intestinal bacteria fall into the facultative aerobes. In other words, again, I don't need oxygen, but if it's there, I'll use it. Staphylococcus is another one. I don't need it, but if it's there, I'll use it. Anaerobes. Okay, so anaerobes, so remember, this is oxygen, right? Anaerobe means without. Okay, so an anaerobic organism doesn't like oxygen. It has to be without it. So why? Remember we talked about those detoxifying enzymes? They lack them. So if you put them in a place with oxygen, they're going to die. Okay? Literally, oxygen becomes these toxic things that I have no way to get rid of. So it kills them. So, examples, a lot of the oral bacteria that get in between your gums and the socket where your teeth are, where there is no oxygen because it's literally in a pocket of tissue. Um, some of the intestinal bacteria, like I said, many of the gram-negative intestinal bacteria are facultative, but some of the intestinal bacteria are actually anaerobes, where if oxygen comes around, they're not going to like it. Now, aerotolerant anaerobes. Aero is oxygen, right? Tolerant. What does it mean to tolerate something? I tolerate my brother, right? Okay. In this case, they are anaerobes, but they're tolerant. They don't use oxygen, but they can survive and grow to a limited extent in the presence of oxygen. Lactobacilli, certain ones of those, certain streptococci, they can actually survive with oxygen um, and grow, but they don't need it. Okay? Sorry, I'm gonna get some water. Okay, so another factor that can influence their growth is the pH, potential hydrogen. pH is the degree of acidity or alkalinity, how basic is it, of a solution, and it is expressed in a scale from 0 to 14. 0 being the most acidic, we're talking like sulfuric acid, hydrochloric acid. 7 being neutral, we're talking about double distilled water. And 14 being the most alkaline or basic, lye. Lye that you use in soap is very, very basic. Most organisms, or actually, I'm sorry, sodium hydroxide. Sodium hydroxide is the most alkaline. Most organisms live between 6 and 8. And the reason for that is because if you get into strong acids or strong bases, it's going to be damaging not only to things like enzymes, but the structure of the cell will also fall apart. Um, acids can melt your skin. So can highly alkaline sodium hydroxide can melt your skin. So it can be very, very damaging. Some organisms can survive in extreme pHs. Hello, we've got the extremophiles, right? So acidophiles, remember, phile means that they love it, right? So acidophiles actually do thrive in acidic environments. A lot of molds and yeasts can tolerate acidic pH. Alkalinophiles, again, phile love, alkaline is the basic, okay? They thrive in alkaline conditions. So an example of a medically important alkalinophile would be Proteus. Proteus can actually create alkaline conditions to neutralize your urine, and when they do that, it creates the perfect environment inside the bladder for them to grow because they've changed the pH so much. Again, they are alkalinophiles. They want it at a higher pH where it's more basic, where, yeah, they their window is to thrive. Neutrophiles. So remember I said you got neutral? They're actually going to live between the pHs of 6 and 8. Most organisms live there. Another good example of an acidophile, if you've ever seen mold on an orange, any type of citrus is going to be acidic. That would be something that would be an acidophile. So, osmotic pressure. Anytime you hear osmo, think water. Okay? So, most microbes prefer to live in a hypotonic solution, isotonic, or a hypertonic. So hypotonic means you have more water than salt. So it's kind of a less salty solution. In an isotonic, you basically have equal parts of salt and water. If we're talking about salty, then that would be hypertonic. It's got a lot of salt and very little water. So, osmophiles, they live in habitats with high solute concentrations. In other words, they like it really, really salty. These are kind of, remember I talked about the bacteria, the extremophiles who live in the salt seas or in the salt lakes. These are going to be the osmophiles. They like it super salty. Osmophiles are what you would find in canned goods. You know, when canned goods go bad and they expand? Well, canned goods have very high salt content to try and keep bacteria out. Well, the osmophiles like it salty, so they be in there. So yeah, if you're ever get a can of something and it looks like it's blown up, don't ever eat it. Okay, halophiles. They also live in habitats with a high salt concentration. Okay, obligate halophiles must live in saline environments. Again, obligate. They're obligated to be there. Facultative halophiles. Again, facilitate. I'll help it along. They tolerate high salt concentrations, but they do not necessarily have to live in the saline and the salty environment. Staphylococcus aureus is actually a very important pathogenic halophile. Okay. Usually it's normally in our nasal cavity. You know, because you've tasted mucus, don't act like you haven't, that it's salty. Okay. So the problem comes when it gets into the wrong place. So we've got Staphylococcus aureus in our nose 24/7. It's always there. But Staph aureus can basically take advantage if it gets into a wound that's open already or it gets into the wrong place. And that's when Staph aureus can get dicey. Okay. Um, Radiation. Phototrophs will use um, visible light rays as an energy source. You know that if you go out into the sun, in bright sun, your skin heats up. That's actually the light rays coming down and depositing energy onto your skin. Non photosynthetic microbes tend to be damaged by toxic oxygen products that are produced by contact with light. Okay. So, UV radiation, again, sun, can cause things like mutations in your DNA. It can damage most types of microbes. Basically, it can be a really good way to control microbial growth. You guys have probably seen on TikTok or on YouTube products that you can't live without from Amazon. And some of them are little cases for your phone that have a UV light. Some of them are little tops for your toothbrush that have a UV light. And the whole reason for that is because of this, that UV light damages microbes. In fact, in the micro lab, in this lab that I am currently in, we have a box that has all of the goggles for different experiments. And when you guys finish with those goggles, I turn on a UV light and expose those goggles to UV light. So if there are any microbes, they die. Why? Because UV causes mutations in their DNA. And it's so damaging that it'll kill them. So, one of the reasons that when we go out, and anybody who's Caucasian goes out into the sun, that we put sunscreen on is because of this. My dad is a white guy. I'm a white guy. And when he was little, he used to go out and spend all day out in the sun hunting squirrels and doing whatever because my dad was my dad. But now, as he is older, he has to go every 6 months to get his skin checked by a dermatologist because he gets skin cancers. And the damage that the sun caused to his skin caught up. So, people that are out in the sun all day, they're damaging their cells. I mean, that's just fact. And it's because the UV causes the mutations that I'm talking about. Wait a minute. Okay, so relationships with other organisms. Okay, microbes don't live in isolation. They're everywhere. They're ubiquitous. Okay, um, they and often have multiple types of relationships with other organisms in the vicinity. So you're not going to basically have this environment where all you see is a microbe. It just doesn't work that way. So um, they have relationships with the things in the environment. So, symbiosis, two organisms living together in a close partnership. There are three types of symbiotic relationships. Now, I said partnership, right? So, mutualism. It's an obligatory relationship that both organisms benefit from. We have bacteria in our gut that benefit from eating what's in our gut as it's going through, and we benefit because their waste products are usually vitamins that we need. So, they're there, but we benefit from them being there. They're there and they benefit from being there because they're getting nutrients from us as well. That is mutualism. They don't hurt me, I don't hurt them. Okay? Now, commensalism. One partner, the commensal, benefits, which the other is neither harmed nor helped. Um, the best way that I can think, or the best, I guess, way to describe it I can think of is cattle egrets. These are the white birds that you see around cattle. Cattle have flies around them all the time, right? Especially when they're in herds. So there are a lot of flies around. Cattle egrets benefit from being around the cows in the fact that um, they eat the flies, but the cows don't care about the flies. And the flies aren't hurting the cows. But the egret doesn't hurt the cow either. And the cow isn't really getting a huge benefit from it. So, because they're not getting benefit, the cattle egret is, this would be more commensalism. Okay, it would be a good example of that. Now, parasitism. A host organism is harmed at the expense of the parasite obtaining nutrients and a habitat from the host. So, parasitism, tapeworms. So, my grandmother, Southern lady, dad's mom, um, she used to talk about when she was little, used to be able to go and get a pill, and it was a chocolate pill. That was what she called it, and it would help you to lose weight. Come to find out that that chocolate pill had a tapeworm in it. And the reason you lost weight was because the tapeworm was in your intestine, going through all your food, and you weren't getting the nutrients out of the food. That is a parasitic relationship. I guess you could say that as somebody who wants to lose weight, you're losing weight, so that's beneficial. But honestly, that tapeworm does more damage than it does good in that case. Tapeworms can migrate from your intestine. Tapeworms are taking nutrients out of your body. It's a parasite. You are not really getting a benefit from a tapeworm being there. All right. Antagonism. It's an association between free living species that arises when the members of a community compete. Okay? So, the first microbe in an area has a competitive advantage for available space and nutrients. Okay? Um, Antibiosis is the production of inhibitory compounds, like antibiotics, into the surrounding environment to inhibit the growth of other microbes. So, the microbes that are in your intestine kind of consider it their territory. And to a certain extent, and you know it's not always because everybody's probably had a stomach bug, but to a certain extent, when other microbes try to invade that space, they will defend it because it's their space. So there is competition that happens. Now, if the invading microbe is stronger, it'll take over and then you get a stomach bug. But the fact that those microbes are there means that there is competition for that space. Okay. So that's going to be kind of antagonism. Now, antibiosis, anti against bio life. This is things like that fungus that I talked about on the plate, creating antibiotics to clear a space on that agar so that they could eat the agar and the bacteria weren't competing. Synergism. It's a relationship between two organisms that benefits both organisms, but is not necessarily for survival. It's more cooperative effort to produce a result that individuals couldn't really do alone. Okay, so it's not necessary, but it's beneficial. All right. So, planktonic versus biofilms, going it alone versus a group effort. So, free living bacteria that live alone, let's say as a single cell in the big wide world, are called planktonic. But groups of microbes that attach to a surface and form a community are called a biofilm. So, a biofilm starts with who we call a pioneer colonizer. It's the first thing that initially attaches to the surface. So it's the first bacteria that attaches to the surface. Okay. Um, Other microbes will eventually join that group, and they're kind of held together by a matrix, by a goo, secreted by that community. So you've got the pioneer bacteria, but as the pioneers secrete that extracellular material, helps keep them on the surface, it serves kind of as an attachment point for other or later colonizers. A quorum is basically sensing chemicals that are released by the bacteria. So it's kind of chemotactic. It's basically chemicals to say, hey, we're here if you want to come. Members of the community communicate via quorum sensing. Quorum. Signaling, it's a signaling system used by microbes in that biofilm, in that place where they've attached, that help to coordinate their efforts. I want other bacteria to come as well because I want this to get bigger so I'm safer. In many, but not all biofilms, other species will join in. They may contribute to the extracellular matrix and or participate in the quorum sensing with their own chemicals or ones released by other species. So here I've got my original kind of purple bacillus. I've got pink bacillus. I've got red coccus, and I've got blue coccus. So now I've got a group, okay? There is strength in numbers. Biofilms are a lot harder to kill than individual bacteria. This is problematic in hospitals. It really is. Especially with indwelling devices, things like respirator tubes or catheter tubes, things that actually sit inside of somebody's body. This is an issue, okay? Biofilms serve as a constant source of bacteria that escape. Literally, this biofilm is releasing bacteria and they will become free living again. And which one you get, who knows, especially if it's a big mixture, if it's a big group. You may get 2, you may get 4, you may get 1, but you never know. And you never know what kind, because if it's a mixture like what we have here, you don't know if you're going to get, let's say, streptococcus versus staphylococcus versus micrococcus luteus, whatever. Okay. All right.